Okay. All right. Thank you for joining us today. We have an exciting lineup of speakers to introduce EMSL's latest project, the Molecular Observations Network, also known as MONet. Before we get started, I wanted to ask our in-person attendees to please silence your cell phones, avoid, if possible, moving chairs, and resist the urge for some silent side conversations. We're broadcasting this live, and we want to make sure that our virtual audience can hear our speakers clearly. Also, thank you to our virtual audience for tuning in. If you have questions as this event proceeds, please add them to the live stream chat. We'll be gathering your questions and answering as many of them as we can at the end of this program. And with that, I want to introduce Dr. Steve Ashby, the lab director of the Pacific Northwest National Laboratory.
Well, good afternoon and welcome, everyone. Today is an exciting day as we come together at PNNL's Energy Sciences Center to launch MONet, short for Molecular Observation Network. We're honored to be joined by DOE Office of Science leadership for this special event. On behalf of all of us at PNNL, I extend our deep appreciation to you, Dr. Berhe, and your team for your engagement this afternoon, as well as the time you spent with us over the past day and a half to meet our talented staff and to witness firsthand the breadth and impact of their contributions. MONet is one of the ambitious science objectives foundational to the 10-year strategy for the Environmental Molecular Sciences Laboratory, or EMSL, which we proudly steward on behalf of the DOE Office of Science. I commend EMSL Director Douglas Manns and his team for developing this exciting strategy. The MONet platform is more than 2 years in the making and the personal passion of many of you in this room. Through your individual contributions and close collaboration, you are bringing an unparalleled research capability to our national and international scientific community. MONet will help to address a critical gap in our understanding of the molecular, chemical, and biological level composition, structure, and interactions in soil systems and the plant and microbial communities living within. It also will aid researchers seeking entirely new insights into how these soil systems perform important functions, including the uptake or release of greenhouse gases and the cycling of carbon, which in turn will improve multi-scale models of Earth systems across entire regions. Today, we'll hear from our MONet core team, who will explain how it works, the data we will obtain, and the value it will bring to understanding ecosystem function and response. I wish to take a moment to acknowledge this core team, comprised of John Bargar, Emily Graham, Sarah Leichte, Corilo, Maggie Bowman, and Odita Chafuku. I hope I got all those names right. And of course, something of this magnitude requires the talents of many more people, and in this case, the contributions of more than 30 of our colleagues in EMSL, where their collective expertise embodies a multidisciplinary approach to addressing national and global challenges that is a hallmark of a national laboratory. And we could not be prouder of the efforts that you've expended to bring us to this moment in time, and we eagerly await your future discoveries. Now, I know we're all eager to hear from Dr. Berhe, so it's my honor to introduce her today. Dr. Asmeret Berhe is the director of DOE's Office of Science, which is the largest funder of physical sciences research in the United States. Key to its success is stewardship of the world's largest array of scientific user facilities, including EMSL and the Atmospheric Radiation Measurement Climate Research Facility, both of which PNNL proudly operate on behalf of DOE. In her role, Dr. Berhe also provides oversight for the 10 national laboratories stewarded by the Office of Science, and this includes PNNL. Dr. Berhe is currently on leave from the University of California, Merced, where she holds the Ted and Jan Velasco Chair in Earth Sciences and Geology, is a professor of soil biogeochemistry, and previously served as the Associate Dean for Graduate Education. Dr. Berhe's internationally recognized research lies at the intersection of soil science, global change science, and political ecology, with an emphasis on how the soil system regulates the Earth's climate and the dynamic two-way relationship between the natural environment and human societies. She previously served as chair of the U.S. National Committee on Soil Science and a member of the Board of International Scientific Organizations at the National Academies, among other prestigious positions. Her scholarship and efforts to ensure equity and inclusion of people from all walks of life in the scientific enterprise have received numerous awards and honors. And as many of you know, Dr. Berhe has been an active EMSL user. We had to tear her away from her tours in EMSL just a few minutes ago to get here, dating back more than a decade, and most recently co-authored a paper in 2022 with colleagues in EMSL regarding the impacts and mitigation strategies for forest fires on soil ecosystems. She's clearly a hands-on leader. So Dr. Berhe, it is our pleasure to hear from you now, so please join me in a warm welcome for her.
Nice to be in the room with a lot of familiar faces. Thank you, everyone. Let me start by saying thank you to Steve for that kind introduction. It's truly a pleasure for me to be back here at PNNL. Thank you so much for inviting me to be part of this incredible event. I could not be more excited about what you're doing here and the inspiring and important scientific endeavors that you're launching today. The research obviously being done as part of MONet is literally in my backyard, right, with all the puns intended there. For those who don't know me, but you heard a little brief introduction from Steve, I'm a soil scientist, a biogeochemist by training. Of course, it's not lost on me that I'm also the very first earth scientist to be serving in the position of director of the Office of Science. I've been in this position now for a bit less than a year. And as you heard, I was previously a professor. Well, I'm on leave from my position as a professor of soil biogeochemistry at UC Merced. And so I could not be more at home with the research that you're all doing, the mission and the goals of this project. Of course, I think I should note that when I was a grad student, when I first came to EMSL and PNNL to conduct research, that it would not, have occurred to me that I would be returning in this capacity to help get the word out about a tremendous new opportunity that we're here to celebrate. So when I was a grad student at Berkeley, I was very fortunate to be able to be advised and mentored by some folks' names who's very familiar to people doing this kind of work and folks in the room. So I had my advisor at UC Berkeley, John Hart, who was a pioneering ecologist. He was actually a converted, reformed physicist, if you will, who decided to do global change work. But at the same time, I had two co-mentors. One of them was at the Lawrence Berkeley National Lab, Margaret Thorne, and then another one, Jennifer Harden, who is at USGS Menlo Park. And it's really the mentorship that I received, in particular at LBL by Margaret, that was the reason why I was able to even know about EMSL and was able to write user proposals to come and visit and spend time at PNNL. And I should note that along with those visits, I spent a lot of, quote unquote, fun times at the guest houses while I was working around the clock on EMSL. But in all seriousness, though, EMSL and PNNL were instrumental in the success of my research and helping us really advance the work that we were doing at the time, which was to help improve our understanding of how geomorphic processes control the soil's ability to sequester atmospheric carbon dioxide. We were able to use NMR to determine the composition of soil organic matter across a watershed in the first project that we did. And so specifically, we tried at that time and successfully applied provide both direct polarization and cross-polarization, in particular CP magic angle spinning 13C NMR techniques in soil along both topographic and depth gradients so that we can demonstrate how and why soils in dynamic landscapes, in particular in the depositional landform positions, were able to not just store but also stabilize carbon and organic matter. And so, to be able to do this work, though, as a student with my kind of background, I came into the project with limited coursework in spectroscopy, right? Because it simply wasn't available to students pursuing topics like I was studying at the time, especially. So I pretty much learned everything that I needed to know about the spectroscopy and got hands-on experience that I was able to apply through my entire career, really, including application of that work through some of my students that is happening right now by working very closely with scientists that are here at PNNL and EMSL. I think, for example, I'll give a lot of credit, for example, to a person that you all might know, Sarah Burton, who works at EMSL, and others who were associated with EMSL at the time, and the NMR groups in particular, for the many lessons and collaborations that they were able, kind of to generously share with me over the years that have made better not just my own research, but also the research of my mentees and collaborators across the many years we've been working with EMSL. And kind of switching gears on the topic of today, I love this idea behind the MONet project, not only because I care deeply about soil, but because this research also has so many components that make it exemplary. It includes, of course, fundamental discovery science that we will learn a lot from this, but it also includes a component that's inclusive citizen science kind of project. And it also allows us to make the hidden world visible, right, in many ways, connecting the local processes to national ones. And it also then in the process keeps making contributions to how we can solve perhaps part of the climate crisis. And one of the aspects that I like most about MONet, it also has to be that it is clear. It has this really neat, well thought out plan. And as many of you here would know, MONet was established as one of EMSL's 10-year objectives. But before the project could be launched as a full-scale program, researchers started with a much smaller pilot project that we got to hear a little bit about today. And this pilot that was called 1,000 Soils aimed to collect, create, and analyze these samples coarse across the United States, and we'll do so in the project too, to help understand many of these processes in soil. And it's incredible what Emiley, the earth scientist leading this work, Emily Graham, and the other researchers in these projects have developed and have enabled to be standardized for the collection so that the workflows can be standardized for soil samples. And I also have to admire how they've worked and continue to work with indigenous communities gathering this data. And that from the get-go, the project was able to engage communities, and in particular communities that we must, all of us in science, do better to serve and engage in the work that I do. I think this is extremely important. It has huge value in understanding soil systems and improving our understanding to predict climate impacts and many other issues. So our Office of Science, these outstanding user facilities that we have like EMSL are incredible because they allow us to have these really important, they could play important roles, if you will, in efforts like this. And the ambitious EMSL MONet project and its objectives show what can be accomplished when we work together, bring our heads together, right? And so I like to take a moment to chime in about our amazing user facilities overall. Of course, as you heard, there's 28 of them that we have at the Office of Science, including EMSL. And these user facilities are, of course, homes to the most advanced tools across many areas of modern science, and ranging from, supercomputers to particle accelerators to specialized facilities for things like nanoscience and genomics and more. And of course, they include EMSL. And together, these tools that we have in their user facilities provide the scientific community with critical and unique, I think it should also be said, premier capabilities for addressing so many of the modern challenges we're trying to work on these days, including in areas of energy and environment, and areas that have to do with some security challenges and more. And as a user facility then in EMSL, MONet would provide leadership in fundamental biological and environmental sciences. And the scientific community that can then leverage EMSL's world-class scientists, biologists, earth scientists, chemists, and computational engineers and software developers, and many of these state-of-the-art tools and instruments and workflows, including high-performance computing that includes powerful software tools that can improve efficiencies in not just collection of data, but how we capture it, store it, analyze it, and more. And then researchers from around the world can hopefully get a chance to access these premier tools, not just here at EMSL, but also at all user facilities at no charge, as we always do. And of course, once their research proposals are accepted through a competitive selection process. And now with this and so many of the other things that we have seen today, for example, EMSL has made a giant leap forward in a breathtaking and engaging way in developing MONet, and such an exciting, of course, new national collaborative for addressing challenges in soil science research. The molecular observation network will provide, of course, molecular level information and microstructural information on soils, soil water, soil microbial communities from soils around the U.S. through a robust, free, and completely accessible databases. I can't tell you how happy that makes me. This is incredible. Soils are amazing, but they're also complex and not necessarily the easiest things to study. So these kinds of things make a world of difference. And through a growing range of partners like the Joint Genome Institute and the National Ecological Observatory Network, or NEON, who are also national user facilities in their own right? MONet seeks to work with federally funded labs, academics, students, and citizen scientists in this incredibly ambitious and critical, important project. And the collective work, I think, that's being done here will further our understanding, of course, of some really key basic soil system processes throughout the country and in diverse environments, enrolling and enabling scientific discovery that I think that will be conducted by all, but also has a benefit for all. The idea of engaging citizen scientists in these kinds of collective efforts in particular, I think, are just incredibly important. And it really helpful also to bring the public along, if you will, as we unlock the mysteries of the molecular world, literally that's under our feet. So, it's really super cool about MONet is that I love the fact that anyone can submit their samples. Anyone can volunteer to be part of this. And the goal to make this completely accessible to wider research, kind of world that's out there is just amazing. Everyone, I think, when we engage people in research in one way or another, especially students, I think we see the benefits of that in many different ways. So many people can benefit, and we want publicly funded science, of course, to benefit everyone. And so this is extremely important. And of course, this is different, right, from how many of the other user facilities, which require competitive proposal process for use, function. And that this new direction, I think, with MONet is amazing, and it would engage, hopefully, more people. It will be inclusive, and let really anyone to be able to dive in today to ask communities and researchers from across the country, literally to be part of this and contribute and share a piece of their backyards for the science and many other benefits that will come from it. So I don't think it would not be a surprise for you all to hear that I think this is extremely exciting and important work, and I think we simply just don't have enough understanding and detail and data on how soil processes operate. Things like respiration, for example, how do they occur? Why do they vary from one place to another in different types of soil? And how do we represent things like this in our efforts in earth system science and the modeling capabilities that we heard about earlier, for example, with E3M and others. And so being able to collect these kinds of molecular data that MONet will capture will be incredibly important in helping us improve our understanding, capabilities to model it, and even just better insights into the models that would help us understand soil processes better. And so, the citizen scientists, the students, members of the academic community and industry, folks working in our national labs, other researchers, I think everybody will be able to benefit from what will be derived from this project. And since it's also open, it could be the perfect project, right? For, you can imagine, junior high school student, like my kids are at this point, university students, graduate students, of course, and others that could get involved. I can imagine youth groups in every corner of the country and families, and may potentially even getting their hands on some kits and contributing to this incredible effort. And from what I understand, you'll also be sending folks these giant soil kits in a suitcase that has all the standard sampling kits that typically, only soil scientists get to play along with that, to play with toys like that. And I think everybody should have a chance to do that. I think soil is cool, in case that's not clear so far. So I feel like it's really a good idea if everybody gets a chance to get their hand dirty and learn more. And of course, researchers can use the equipment in that soil kit that they're sending them to dig the scores, give them the info they want, and contribute to the metadata EMSL needs. And once the samples are then sent back here to EMSL, they will run through their kind of list of analysis that they've laid out. And once the information then is collected, be able to join the database and be freely accessible to the public. I think this is exactly how publicly funded science should work. Right, publicly funded science that we enable through the Office of Science, for example, through our many of our programs, including the Biological and Environmental Research Program, is exemplary of what kind of what we can do together. And we're doing this for not just for now, but also for the future and the future health of our environment, and it's everybody's future that we're looking to protect, of course, and inform. We must turn to fundamental science, of course, to figure out what is driving climate and climate-related processes. So this is a call to get everyone involved to contribute to the soil story of their community, doing this for now, for their future, doing our part to mitigate the climate crisis. And as we always, as we do, we're looking to the fundamental science for solutions. And I applaud you and everyone involved here on this milestone that you've reached at this point, knowing we have, obviously, of course, a lot of work left to do, a long way to go on this fundamental science journey that you're all embarking together. So thank you, everyone. I really appreciate you all inviting me and being part of this. Thanks.
Thank you, Dr. Berhe. For this next portion of the event, you hear from our MONet researcher team on the various analyses that we'll perform on these soil samples and how that data will be analyzed, captured, and made available for everyone to use. I'm honored to introduce MONet's lead scientist and honestly the heart of this project, Dr. John Bargar.
Thank you very much, Douglas, and what she just said. So soils contain enormous amounts of carbon, more than all the above-ground biomass around the planet and all the CO2 in the atmosphere combined. Moreover, soils are intimately in contact with the atmosphere, and they interconvert carbon between CO2 organic and mineral forms. This means that CO2 can escape from soils, and it also means it can be stabilized in soils. This means that soils are profoundly important to controlling atmospheric CO2. I just want to point out that soils contain both inorganic and organic forms of carbon. Today we're focusing mostly on soil organic matter, which forms the largest pool globally. So whether or not carbon will be stabilized in soil or will be released back to the atmosphere as CO2 or methane is determined by molecular processes that govern the movement of carbon between plants, microbes, and minerals. And these processes are highly complex. Imagine, if you will, a large organic carbon molecule that breaks into lots of smaller molecules, each of which can continue to break down into lots more smaller molecules, or can stick onto mineral surfaces, or can dissolve into water, or can be eaten by microbes, in which case it all goes around the circle again. There's a lot going on here. To make matters more complicated, the rates of all these processes are highly variable. Very few models incorporate the needed molecularly specific information, but they should, because this process complexity leads to uncertainty in Earth systems and climate models. Indeed, our failure to represent this process complexity, this molecular process complexity, is one important reason why atmospheric CO2 predictions contain so much uncertainty. We urgently need to do a better job of understanding these molecular and microscale processes. But we also need to understand these tiny processes over very large scales like you see here. This is what MONet is about. And here's why. As you look out across this landscape, you can see lots of individual soil plots, and you can appreciate that each plot receives different amounts of sunshine or rainfall, has different vegetation, different land use types, blah, blah, blah. I could go on. The main point here is that each of these soil plots will respond to climate change or to drought or to flooding or other types of forcing events in very different ways. Our job is to integrate all these different types of behaviors and how they interact with the atmosphere together to understand how climate change is acting. That is to say, we need to collect soil samples across entire regions, which brings me back to the statement I made just a minute ago, which is that we need to understand these tiny processes over very large length scales. So how do we do this? We use models to connect across distance and time, but models are no good without data. The problem is that we don't actually have standardized molecular and microscale data across entire regions. So this is so important that I'm going to repeat it. We don't have standardized molecular and microscale data across soil regions. But thankfully, we do have soil ecological networks like NEON and Ameriflux that are collecting and compiling soil data across the United States. These networks aren't focusing on molecular, carbon, or microscale soil properties. So the MONet program is creating a new database to provide this critical molecular and microscale soil data, and also to build on these other ecological networks. So here's how we're doing this. The most important product is a high-quality, high-volume database of these important information resources that are needed by environmental and climate scientists, modelers, and modelers and experimenters alike. The data need to be findable, accessible, and searchable by everyone. That is to say, we need to ensure that access to these data is democratized for all communities and scientists to use it. Dr. Yuri Corilo will talk more about this in just a few minutes. To generate big data, we're finding ways to convert traditional labor-intensive molecular methods into high-throughput standardized workflows. So standardization is a term you'll hear a lot about today. There will be a quiz at the end, so everyone take notes. Standardization is important because it's critically important. It ensures that all of the data will be usable. It also reduces uncertainties within the data, and it also helps remove the effects of inherent human bias. So within this molecular space, the Joint Genome Institute, or JGI, is a very important partner. JGI is supplying critical molecular data on soil microbiomes that are going into the MONet database. So Dr. Emiley Eloe-Fadrosh from JGI will be, along with Dr. Tomasz Varga from EMSL, will be speaking in just a few minutes more about molecular methods. Before we can create big data, however, we need to collect thousands of soil samples from across the United States. To do this, we developed a sample collection kit and standardized procedures for scientists participating in this open science network. We send these kits to scientists across the country. They send them back, we analyze the data, and provide them with results. In this endeavor, NEON has been a very important partner. Last year, during a pilot project, NEON collected and sent us more than 70 cores, which were extremely important in helping us learn how to do this important step. So Dr. Maggie Bowman from EMSL will tell us more about our standardized soil collection procedures in just a minute. So this is a very big project. We simply can't do it ourselves because it's just simply too big. However, by collaborating with thousands of partners across the United States, we can solve truly large problems together. In case you haven't noticed, this is not a traditional way of doing science. MONet is an open science network. Open networks provide a way to solve truly challenging problems at scale. And we're providing you with an opportunity to participate in this open science network. There's no cost to participate other than collecting and submitting the sample cores. As I mentioned just a minute ago, we analyze the samples and provide you with more than 20 advanced data types for your research. Moreover, these data are accumulating in a cutting-edge database that allow anybody to access it, so it's win-win-win on all sides. Crucially, also, open networks provide opportunities to expand participation by traditionally underrepresented communities. Along these lines, we're inviting undergrad as well as graduate students, postdocs, faculty from schools across the country to participate. We're also inviting high school teachers, national lab researchers, industrial scientists, citizen scientists to send us soil cores. So by growing new collaborators, new data types, and a new user community, we'll transform our understanding of soils and provide new tools to society for adapting to and counteracting climate change. And with that, I'll stop, say thank you very much, and turn the baton over to Dr. Maggie Bowman, who will tell us more about our standardized soil collection.
Hi, thank you. As John mentioned, I'm Maggie Bowman, and we have designed a standardized protocol and sampling kit, which includes field measurements and a soil kit. And Dr. Berhe mentioned it's a large suitcase that is shipped to participants to collect samples. These standardized field measurements and protocols are used to reduce variability. This will help us overcome and compare measurements across diverse soil types, disturbance, and ecoclimates. When a MONet proposal is accepted, our team starts by scheduling a meeting with you to coordinate a sampling plan. Then we'll ship the soil sampling kits, coolers, and everything you need for sample collection directly to you. The sampling kit contains all the equipment you would need to collect soil cores, as well as alternative methods for stubborn soils. You will collect 2 large cores, which typically contain the organic and mineral soil horizons, as well as 4 smaller cores to make sure that we capture the representative diversity of your surface soils. You will also collect field measurements like temperature and soil moisture. Once your cores are collected, you will send your kits back to us for immediate processing using our standardized protocols. When soils arrive at the lab, your cores are unpacked and we verify that all information is complete. The 2 large cores are used for 2 main workflows. One is used for time-sensitive measurements, where the other is used for structural measurements. The core used for time-sensitive measurements is processed immediately while the soils are still fresh. That helps us characterize the biological and chemical composition of the soil. For this core, we collect measurements ranging from the molecular to bulk scale. And our molecular measurements include data like metagenomics and high-resolution organic matter composition, while the bulk characterization includes data like respiration and total carbon. The core used for structural measurements allows us to capture data on the physical structures of soil. This includes high-resolution X-ray computed tomography on the intact core and coarser scale measurements like texture at different depths. Now, I really want to highlight again the reason why this standardization is so important. It's because we're measuring very, very tiny things, measured thousands of times across hundreds of soils. If this collection, process, sample handling, analyses are not standardized, then the pieces won't fit together in our great puzzle. With this, Tomas Varga will provide an example of some of the very tiny things we are measuring and how we can use that data.
Hi, I'm Tomas Varga. Let's talk about the very tiny things we are measuring. EMSL uses a highly sensitive and extremely powerful technique called Fourier transform ion cyclotron resonance mass spectrometry, or FTICR mass spectrometry. It is a unique and powerful tool in helping us characterize soil organic matter. There are only a few of these instruments in the United States, and our 21 Tesla magnet makes it one of only 2 in the world. So EMSL's fleet of mass spectrometers is a national resource for soil organic matter characterization and modeling. From our ultra-high resolution FTICR mass spectrometry, we got an unparalleled molecular scale view of the chemical complexity of soil organic matter. With FTICR mass spectrometry, we are able to detect and determine the chemical formula for thousands of molecules in soil samples. This helps us understand the types of organic and inorganic chemical transformations that occur in different soils across spatial and temporal scales. The connected open spaces that exist in soils, what we call soil pore networks, direct the flow and transport of minerals and microbes in the soil. This influences carbon respiration, soil organic matter decomposition, and eventually carbon dioxide and methane release. Therefore, in addition to the molecular information, we also gather structural information about soils. Using X-ray computed tomography, we gather high-resolution data showing how the different soils are packed together. In other words, we look at where open spaces exist in the soil, again called soil pores, and we look at how those pores are connected or isolated. The data from X-ray computed tomography helps us create a 3-dimensional image of each soil core's internal pore structure. Through these 3D images, we can determine pore sizes, their distribution, and pore network connectivity. This 3-dimensional rendering of a soil sample is from a micro-computed tomography scan. Here we are looking at the inner pore structure of a soil sample. The unconnected pores are shown in red, and the connected pores are shown in blue. We can also see how the pores are connected, the pore network, through the white lines and yellow dots. Through this level of detail, we can see the many factors that control organic matter stability in soils. That is how easily it can be converted into carbon dioxide or methane. Organic matter can be protected in the smallest pores so small that you can't see them in this image. Organic matter can also be protected by physically or chemically binding to mineral surfaces. Soils with very small pores tend to have lower permeability, meaning that water doesn't move through as easily through the soil as easily as in soils with larger pores. Low permeability tends to create chemical conditions where organic matter decomposition is slow. I mentioned these factors to demonstrate that understanding the chemistry of organic matter is crucial to understanding its decomposition. As John noted earlier, we can reduce uncertainty in Earth system and climate models by including more chemical specificity. And this is where FTICR mass spectrometry is important. It can provide us with that level of chemical specificity. Now, with additional experiments and FTICR measurements, we can actually learn what molecules are contained in various size pores. And while we are on the topic of soil pores, Emiley Eloe-Fadrosh is here to discuss how JGI will conduct metagenomic analysis of the microbes contained in these pores.
Great. So there are more microbes in a teaspoon of soil than there are people on the earth. But studying the diversity and function of these microbes is really challenging using traditional microbiological approaches. We cannot grow them in the laboratory. We cannot do experiments using controlled conditions to understand how they decompose this organic matter, how they interact with one another. Instead, we use a method called metagenomics to access their genomes in bulk and compare these sequences to what we know about the tree of life. Metagenomics is a lens through which we can access wild microbial genomes and viral genomes to infer function and to get a clearer picture of who is out there, what are they doing, and how they're impacting climate. Through an innovative partnership with EMSL, the Joint Genome Institute will support large-scale metagenome sequencing for the MONet program. The JGI is a flagship DOE user facility that provides genomic resources to the global scientific community. Over the last 25 years, the JGI has pioneered metagenome sequencing and analysis approaches from diverse environmental samples. Our team at the JGI has enabled remarkable science by harnessing our metagenomics expertise to uncover novel microbial groups and viruses from soils, from oceans, from plants and animals, and deep within the Earth's crust. We have scaled sequencing 30 million-fold through the application of new sequencing technologies like the Illumina NovaSeq and developing new computational tools to assemble and annotate this immense data. By partnering with EMSL and the research community, the JGI will generate several hundred metagenomes on an annual basis, which amounts to nearly 20 terabases of data per year from diverse soil microbial communities. This is just an incredible scale of data from soil microbiomes. The JGI will also use our state-of-the-art analysis pipelines to process this data, taking a genome-centric approach to the assembly and annotation and providing a large collection of wild microbial and viral genomes from soils. The JGI will also host this treasure trove of metagenome data in the Integrated Microbial Genomes and Microbiomes computational platform, along with seamlessly sharing this data back to EMSL. The standardized pipelines allow data to be compared across these diverse soils and will allow the research community to study how soils compare to one another across field sites and across the United States at scale. And of course, this metagenome data can be analyzed within the context of all the additional molecular measurements that you've already heard about to gain a really comprehensive picture of soil microbial communities. Beyond the tools that JGI and EMSL will provide for metagenome data processing and annotation, I'm really excited to think about how new tools like machine learning can be applied to gain even more insight into microbial function. The MONet metagenome data and all the rich molecular measurements will be such a foundational reference catalog that will enable new science to be done, from modeling microbial communities to understanding their function, to being able to predict how those communities will change or be resilient over time. I can also envision how this remarkable data could feed into new climate models that incorporate microbial contributions in a way that is just not possible right now. Now I'd like to hand it over to Emily Graham to share some initial insights on the Thousand Soils project.
Thank you. So I'm Emily Graham. I helped lead the Thousand Soils pilot. That helped us develop standardized methods and approaches for MONet. I'm always super impressed by all my colleagues, and I think what you've seen so far really highlights that EMSL's molecular observation network can give us unprecedented insight into the mechanisms that regulate key environmental processes. So one thing that people always ask me is, okay, so now you have all this ultra high resolution data. What do you do with that? And so that's something we're really focused on right now, using our data from the Thousand Soils pilot, helping develop example use cases and tools for our user community. So during that pilot effort, we were able to collect nearly 100 sets of cores distributed across the continental US. These covered a wide range of variation in environmental parameters that we know are associated with soil carbon cycling. So things like moisture content, carbon and nitrogen ratios, pH, and other factors. And so for a first look at the data, we combined our data from our FTI mass spectrometer that Tomas talked about. Will has been really involved in that as well. And microbial metagenomics that Emily just talked about. We looked at differences in carbon cycling across different biomes with these data. So from the mass spec data, we can pull out inferred biochemical transformations that really tell us a lot about the specific carbon cycling reactions that are associated with respiration. And from metagenomics, that gives us an idea of the full range of metabolic pathways that can possibly occur in a biome. So we can take these two data types and then correlate them with the soil respiration measurements that we take and get an idea of very specific pathways that may correspond to land-atmosphere fluxes of carbon dioxide. So when we do this may not be surprising to you guys, we see a tremendous amount of variation across different biomes. And while this isn't surprising, it's also not typically how we represent carbon cycling in models. So even the most state of science carbon cycling models are really just different pools of carbon that are linked by different processes that connect them. So these models in general are structured to be widely applicable, right? So we don't have to have detailed models for different ecosystems. But that's in fact what the data are starting to show. So just to give one example here, when we look at data sets distributed across all deserts within the continental US, we're seeing that amino acid and manganese cycling may be important and under-considered aspects of carbon cycling. When we put this in the broader context of climate change and changing precipitation regimes, we know that certain areas of the country are going to get drier. And so these may be really important mechanisms to think about in our next generation of models. Some other things that we're doing here, we're using machine learning and process-based models to look at specific processes that may be important as well. So to give one example, from our FTI-CR-MS, we get 10,000 or more different molecules per samples. A lot of those are inactive molecules, and only a fraction of them are cycled. And so we were able to pull out 2 mega features from machine learning that correlate strongly with respiration. And so this is a really tractable set of data that we can then put in process-based models like pFlowTran, which is a reactive transport model used by the DOE user community. So interpreting these data, I think, is one of the most pressing soil science and climate change related challenges of our modern day. We're getting a lot of cool things with just a few of us looking at the data, and I am confident we're really just scratching the surface. I can't wait for the broader community to get their hands on this data so we can all put our brains together and start pulling out different trends and processes that we really need to represent in models. And so next up, Yuri is going to talk about how we're managing that data, how we're making it fair and accessible, and how we're encouraging people to be creative with our data.
Thank you, Emily. So, hi, I'm Yuri Corilo, and I'm here to talk about the importance of democratizing the data. So, and how we're going to do our part of democratizing the data through MONet. So, as we know, access to the data is critical for decision-making in both the professional and academic spheres. But limited access to the scientific data can only create barriers to its democratization, and it slows down the pace of scientific innovation. So this is where the importance of having a defined data model comes into play. So you're probably wondering, but what is a data model? What is a defined data model? Well, essentially, it is a model that allows us to organize, document, and define data in a standardized way. So what are the tangible benefits? Well, it makes it easier to access, understand, and reuse the data. We can use the data more efficiently and more effectively. It reduces the errors of the risk of errors and inconsistencies. So, along with a defined data model, having metadata standards also play a crucial role in decreasing the barriers to the data democratization. Metadata standards provide us an essential information about the data, like its origin, purpose, and quality. It helps improve the understanding and the trustworthiness of the data, making it more accessible and usable by a bigger audience. The efficacy of the data reuse is another reason we need to democratize the data. Reusing data can significantly increase its value, especially when it can be used in multiple contexts and for various purposes. Reused data can save time, reduce costs, as researchers will not have to collect new data. And reused data also enables scientists with different perspectives the ability to examine the data, draw new ideas, hypotheses, and conclusions. For example, data from analytical and molecular experiments can significantly support research community and usher new scientific discovery. By leveraging existing data, researchers can save valuable time and resources and avoid the need to conduct redundant experiments. This in turn allows for faster progress in their fields and the ability to focus on new and innovative research. Additionally, data reuse encourages data collaboration, sharing knowledge across the scientific community. With the ability to build on previous results, findings, researchers can make more informed decisions, identify new patterns and relationships, and uncover new insights. So, the democratization of the data and its reuse in the scientific community can lead to a more efficient and effective scientific ecosystem, driving discoveries and advancement in the fields, in all the fields of research. So, this is why MONet is built with the user in mind. MONet provides a user-centered approach to data management and access. This system will become an essential part of our national system, our national scientific data ecosystem, providing a comprehensive and intuitive platform for data access and reuse. MONet will allow users to easily find, access, and reuse data securely and efficiently. With its user-friendly interface and features, MONet will empower the users to work with the data in a way that meets their specific needs and requirements. Our commitment to MONet users makes a game changer in our national data ecosystem, allowing for the democratization of the data and enabling the next generation of scientific discovery and innovation. Thank you.
Thank you, MONet team. That was excellent insight into MONet's ambitions and how it works. We're now going to talk a bit more about the partnerships we're building through MONet. You've heard of a few of our speakers reference the National Science Foundation's user facility called NEON, or the National Ecological Observation Network. Here to talk a bit more about NEON is Kate Thibault, NEON's science lead.
Hi, the National Science Foundation's National Ecological Observatory Network, or NEON, is a continental-scale observatory that shares in a broader mission with MONet, providing open data to advance regional and continental-scale understanding of ecosystem processes and environmental change across the United States. As a user facility, NEON's more than 180 publicly available data products are collected using standardized methods year after year at 81 terrestrial and aquatic field sites. These sites are distributed throughout the 20 distinct ecoclimatic domains of the US, as you see in this map. Here in Richland, Washington, we are about 175 miles east of the tallest instrumented flux tower in the NEON network at Wind River Experimental Forest. So we invite you all to please come visit us. Next, we'll take a virtual walk around the observatory and give you a few more details. So we'll start with our instrumented systems. NEON has flux towers at each terrestrial field site, similar to Ameriflux towers, equipped with different sensors at different levels above the ground that measure many different aspects of the atmosphere, including barometric pressure and carbon dioxide flux. We also have plots for ground and below ground measurements laid out near the tower. And as an example, our soil arrays and through-fall instruments collect data that can be used to study heat flux, biogeochemical cycling, and much more. Our aquatic sites are also fully instrumented to collect data on freshwater ecosystems, from small streams to larger lakes. And there are plenty of other aspects to our instrumented systems, including automated cameras to track plant phenology, groundwater well stations, and really too many to show here. And they're all maintained by our distributed teams of trained field staff, which number over 350 across the country during our peak sampling season. We also have mobile versions of our instrumentation that researchers with funding can request to be deployed at any location, like you see with this controlled burn in Kansas. And now to our observational sampling, and these are data that are collected by our field staff rather than sensors. These staff sample soil and water and a diversity of taxa, including plants, animals, and microbes. And MONet has successfully leveraged NEON's distributed field teams to collect soil samples, as you've heard about, at most of our field sites already using the excellent MONet sampling kits. Each domain also has a central processing facility for sample processing and shipping to external facilities, such as the NEON Biorepository at Arizona State University that you see here. And finally, our airborne observation platform. These are planes equipped with instrument payloads to collect ecological data from the sky. Our Twin Otter aircraft fly low and slow over 100 square kilometer footprint above our sites, collecting high quality, hyperspectral, LiDAR, and camera imagery. And with these remote sensing data, we can generate incredibly high resolution images of the vegetation and topography of our sites. And we do this all during periods of peak greenness. So all of these systems are designed to be connected and work together to provide open data for use and integration with complementary efforts like MONet for tackling the most pressing ecological questions of today, including, are there really a trillion species of microbes? So now I'll hand it over to Emiley Eloe-Fadrosh to give you a bit more information about the Joint Genome Institute. Thank you.
As I mentioned earlier, over the 25 years that JGI has been in operation, we've provided foundational genomic resources to the global scientific community. This includes sequencing, annotation, and analyzing genomes from plants, fungi, microbes, and microbial communities that are relevant to carbon and nutrient cycling and bioenergy, and developing associated tools and services. The JGI has the experience, expertise, and high-throughput capabilities for environmental metagenomics, and we'll be bringing that as part of our partnership with the MONet program. The JGI and EMSL have a long-standing partnership through the FICUS program, and we are very much looking forward to evolving that partnership through the MONet and continuing to benefit the global research community.
Thank you, Emily, and also thank you to Kate and Douglas. So let's take some time to chat more about the importance of collaboration. So first question, why do you think that we need a national scale collaboration for understanding soils? I mean, how will your facility fit into this effort?
So I guess one of the things that I hope has become readily apparent from the prior speakers is that EMSL can't accomplish this on our own. The science is simply too big. The expertise and the diversity of capabilities that it required is much bigger than what EMSL has. And so a core tenet when we were creating this MONet platform, this objective, was that we recognized the need for strategic and resource partners as part of this to be successful. So although EMSL has a very formidable array of analytical capabilities for soils analyses across multiomics, chemical imaging, and molecular composition, one of the greatest strengths of the DOE Office of Science and the broader U.S. government scientific enterprise are the incredible breadth of scientific capabilities and resources that are synergistic with those of EMSL. Bringing these resources together through partnerships exponentially amplifies the impact on scientific knowledge. It'll help us understand our efforts, and it can generate a tremendous wealth of data and content that's content-rich, multimodal, fair, and high quality that would be unimaginable to assemble without these partners coming together and helping us join this.
Is that on? I'll also add to that and completely agree, Douglas, that the JGI and EMSL as user facilities just have this perfect synergy for providing high quality molecular measurements, multi-omics data, sequencing data, and at a throughput that isn't possible for just individual labs. And so I think this is a really exciting partnership that we've had through the FICUS program and that we will continue to have and evolve through the MONet program. I also think that it's really exciting to have partners such as NSF's NEON because the science really doesn't have agency boundaries. And so by being able to support and collaborate through DOE and NSF resources, we can be greater than the sum of just our individual parts.
Well said, Emily. I'll just add that NEON collects an unprecedented diversity of data about the environment and ecology, but at our 81 sites. And of course, we don't collect the molecular composition data and all of the microbial data, although we do collect quite a bit of that as well, that MONet will be delivering. And so NEON is going to contribute a lot of important contextual data about plants and their diversity and productivity, about the land atmosphere, surface atmosphere exchange of greenhouse gases, in a diversity of other variables that will be helpful for interpreting and understanding the dynamics in the soils that MONet discovers. But MONet is also going to be able to get data from many, many more sites than NEON is able to. So we've concentrated in a set of sites, and MONet is going to be able to collect a more limited set of data, but highly complementary in many, many places with the help of professional and community scientists alike. And the National Science Foundation and the Department of Energy did just recently formalize a memorandum of understanding, and I think this partnership really exemplifies the power of working together to solve our big problems that the planet is facing. And so I'm excited to be here. Thank you.
Wonderful. So can you think of similar past or current efforts like MONet? Essentially, what will MONet bring to the scientific community that's different from past efforts?
I'll start with that one. So large-scale research like MONet is implementing is not extremely common because it is extremely difficult to do. It requires a lot of coordination and a lot of resources. In terms of continental scale environmental data, NASA has really led the way with satellite-derived data sets that provide some really helpful parameters like forest structure, canopy greenness, soil moisture. But of course, a lot of ecological processes operate at different scales. And so we really need to understand all of the scales to enable a holistic understanding. So NEON and MONet bring the local scale and regional scale perspectives in their approaches. So our airborne observation platform brings some regional level data that our ground measurements can be linked to for understanding of local processes. And I think that, I've lost my train of thought. Sorry. It's all good.
I can also
Thank you,
I can also jump in there and say, in addition to the great example of NASA, if we look to the oceanographic community, so being able to monitor different processes within the ocean has been a long time impact, and being able to do so in really collaborative way with large research teams. And so being able to monitor viruses, microbes, plankton communities across ocean scales, across ocean currents could be another example. When we think about soil communities, back in 2010, the Earth Microbiome Project was launched, and that was a large crowdsourced community effort to try to globally characterize microbes from various different environments. And so from my perspective, MONet is really kind of an extension of that concept and idea, but at a much larger scale, and having institutional partners which really make the whole process really impactful by being able to have the data available and accessible in a way that individual researchers can't do.
I was also going to add that the key features of MONet that I think are extremely valuable are the standardization, which is of course something that NEON also highly values, and you've heard a lot about today. But this involvement of the community scientists, so that so many more samples can be collected than professional scientists could possibly accomplish. And what it reminds me of, given my research background, is the United States Geological Service breeding bird survey. And this is a standardized survey that's been going on for decades and relies on thousands of community scientists who are willing to learn to identify birds by sight and sound in their communities. And all of those data are collated and made publicly available. And I use those in my professional research to look at the continental scale patterns in bird biodiversity. And so these are the sorts of uses that I am imagining for MONet's data sets.
So I'll keep my answer short since I agree with everything that was said. I think MONet is meant to complement a lot of these efforts. So, NSF's NEON, Ameriflux, which is another program that DOE stewards in BER, the CZNet, the Critical Zone Network. All of these are large-scale ecosystem studies that I think MONet is looking to complement with our molecular to pore scale resolution data. And so the intent is to help accelerate and more efficiently connect into these multi-scale models, allowing us to go from the first principles models all the way up to ecosystem and Earth scale models much more efficiently than we currently can, because we just didn't have access to that type of data.
let's talk about data now. So how do we store it? How are we going to get it? Who's going to have access to it? Can you speak more about that?
I can start. So there's just going to be a tremendous amount of data, and that's where I think the partnerships really come into play, because both EMSL, NEON, and JGI have experience with handling just the sheer volume of data and how that data can be processed in a standardized way. And so that's where I think this, again, this partnership is really important to be able to have that data. In terms of the metagenomic data, we will be having that produced, processed, and made available through JGI platforms. We'll also be sharing that with EMSL, with NEON, and then also with NCBI, which is the primary repository for sequence data. And so by distributing these resources, we enable broader access to all of the outputs of the MONet data.
Great, and that, of course, is the goal of FAIR data, and we're all strong partners in that. NEON has long been a champion of FAIR data, providing, as I said, all of our data products are available either through an online user interface or through an application programming interface, which is extremely powerful for scientists having to use such large and varied data sets. We also have extensive documentation on our website, as MONet, I'm sure, is dutifully preparing for their data, because it is a lot of work to use these data, but not as much as collecting it. So there's, I think, a lot of great opportunities. I think another benefit of partnership is increasing awareness of each other's data sets as well. So not only are we working with MONet, but NEON does have a relationship as well with Emiley's team at the National Microbiome Data Collaborative. And so we're actively working to get NEON's metagenomic data discoverable through their platform as well, so that even more people become aware of all the various resources out there.
Yeah, so, ultimately the MONet data will all reside in a MONet database, and as Kate was intimating at, there will be pointers to other databases for complementary data sets. So the National Microbiome Data Collaborative, the NEON Biorepository will have pointers so that additional data can be findable and accessible and reusable. I guess I'll speak to a little bit about, who's going to use it and how are we going to keep it, et cetera. My hope is, ideally, that the experimentalists and the modelers will use this jointly, so that as we start to build our models, they will start to find holes or where the model needs to be refined, and that will then inform the experimentalist of what should be the next round of experiments to do to fill those gaps. But equally, you could use the data separately. The experimentalists could use that data to drive their next hypothesis to study, or the modelers could use this data in explicitly parameterizing biological functions that they didn't do before. So I see them as complementary and hopefully driving forward model improvements. I will say one of the things that we, as a core principle for MONet when we were creating this program and this objective, was to accelerate science. And one of the ways that you can accelerate science is by getting that data out there as soon as possible. And so one of the principles with this is that all of the MONet data is immediately accessible. There's no data embargo. So once the data has been quality controlled with the JGI's processes and EMSL's processes or NSF NEON's processes, that data is immediately searchable and available. And anyone can come into the MONet database and download the data. We'll have a login ID or an ORCID ID, but that's largely for data attribution and proper citing, but there's no cost. The data is immediately available. And so I think that's the other core tenet of how we're trying to drive open science and engage this, which my last point will be, I think there's a tremendous opportunity given NSF's mission and DOE's mission around STEM education and DOE's workforce development. These data sets are going to be a massive training ground opportunity, if you ask me, in terms of teachers and researchers in developing hypotheses, doing data analysis skills, model generation, et cetera. And so one of the areas that we're looking to in the future is how our partners can work together to form training curricula using these data sets as well.
All right. Well, this is a perfect segue then to the very last question. So we've mentioned this a couple times, but I want to make sure that we really hit home on this topic, and for our virtual audiences to really understand the magnitude of this project. So who can get involved? You know, let's talk a little bit more about that. We've mentioned it, but talk more. Who can get involved? Who, you know, can participate in this project? So.
Short answer. The short answer is everyone. So when we were creating MONet, one of the interesting concepts that I was envisioning with this was this ability for anyone and everyone to be involved, from high schools to community college to college students, all the way up to the high-powered research institutes at the national labs and the academic institutions. And so, you know, we'll have specific areas of call focus for where we might want samples from, but fundamentally, everyone can be involved in this. And we've tried to make it as seamless as possible, as you saw with Maggie and Yuri's talks around the data and the sampling kits. We have a sampling kit. Everything is included in it. We send it to you for free. All the instructions on how to use it are there. We've worked with the NMDC, the National Microbiome Data Collaborative, to leverage their portal, their metadata submission portal for now, to make it as easy as possible. It's a web-based application, so that the barrier to entry is extremely low. And I think because of that, it truly is meant to be an open science capability for the nation to take part in.
Yeah, of course I agree in the sense that everyone is invited to join in and engage with all of these data that we're talking about. You know, they have been designed with initial questions for use by professional scientists to start with, but of course, you know, I think one of the great things about these kinds of projects and open science is that there are many questions that we hope these data will answer that we never even envisioned in the initial phases of the project. So for NEON, a great example of that is, you know, environmental scientists and ecologists, very few were thinking about artificial intelligence methods when NEON was initially envisioned. And of course now it's a whole new world of options to leverage our data as well as our samples. And I think the more that people engage with all of these data and we work together and we develop new tools, we can also make the data more accessible and meaningful to policymakers and to educators and parents and community scientists. And so the more folks get in there, I think the more benefit that we can derive from all of these open data sets. And the more people that are thinking about these data as well will come up with even new and novel questions and methods that we're not envisioning today.
Yeah, I'll just keep this short and say ditto. I agree with all of that. This is for everyone. MONet was designed to be inclusive and in a very purposeful way. So I think it's a really exciting opportunity.
All right. Well, at this point, we are going to transition into the question and answers. So for those folks who have been joining us virtually, we've been carefully making note of the questions that you've been asking, and so we will jump into answering those as well here in the audience. If you have questions, feel free to raise your hand, and then I invite all the rest of the speakers to come up on stage so that we have a good representation of folks who can answer your questions. So we are going to start with on the YouTube live stream. One of the questions that we received is essentially, what is your definition of quote-unquote fresh soil, and how is that going to be standardized? So I believe this question is for Maggie then. Should be on.
Yeah, so fresh is a relative term. Ideally, we would love to grab these, as we pull it out of the ground, but all the soils are shipped back to us within 48 hours. So all of the sampling happens early in the week. It's shipped out by Wednesday, and we get it back. So within 72 hours of collection, we have preserved the metagenomes. And so we standardize it by collecting that data within the 72 hours of the soil being collected and immediately upon arrival at the labs.
Great. I'm going to give that to Rita. All right, we're going to take a question from the audience. Does anybody have any questions out there? Yeah, wonderful.
Oh, yeah.
This is terrific. So I have two questions. One is you're sending, you're sending these kits out all over, from high school students up to experienced researchers, and you're providing instructions, but how are you managing the quality control to make sure that they're actually following the instructions? I remember as an adult, I had instructions how to assemble things at Christmas, and they didn't always turn out right. And the question is, how sensitive is your analysis to the proper collection of the soil sample? And the second question for the data folks is, you're providing a way of sharing the data, but I know, for example, in ARM, they also provide a number of data products that allow for analysis of data. Will you also be doing that to help researchers take advantage of it? Thank you.
Yeah. Who wants to do the standardization?
Yep.
Yeah?
For the kit.
Okay.
So yeah, that's a good question, and a challenge with any citizen science effort. I will say that we have a notes section that things do go wrong. Even with our professional researchers, things go wrong, like you hit a rock and you can't get the full core. And so we have about three to four backup plans, and the kit, when we run into it before, people provide notes, and it's fairly obvious when something doesn't pass a QA/QC. We have alternative laboratory processing structures as well, right? If they mess up the structure, you can still get chemical and biological measurements from it. And then beyond that, we have pretty robust internal QA/QC standards. And so things will throw flags if through the analytic it messes up somewhere along the lines. I will say I've looked at the data a little bit. It's the cleanest data I've ever worked with. I was putting together my AGU talk, and I was like, this can't be right. Like, this is too clean. So I think for now, we have done a really good job, and we definitely have a very strong emphasis on QA/QC.
In addition to a written protocol, we also, thanks to our great communications team, have a video protocol which shows people how to assemble the kits, how to put it together, and how to do the sampling, which is a great tool for the people who have never sampled soil before, never used a slide hammer, which, I mean, admittedly, my first time using a slide hammer was on this project, because I was the soil scientist that leveraged Neon about 5 years ago and got Neon samples from them.
Okay. So for the data question, like along with just the data that we're providing, like the raw data for the instruments, right? We are providing the data products, right? The standardization goes also on how we're processing, how we're generating that data set. In addition to that, we're also providing how all the software that's being generated, right, on how we're processing the data and make it available for the community to reuse that in different levels of access, right? On the code level, we have command line interfaces. And you also have an API will allow people to reprocess in the way that they would like, right? And connecting those services together to make the ecosystem of their services that we're talking about.
All right. So this question comes from, again, one of our members in the virtual audience. So exciting opportunity. Yes, it very much is. Do you accept soil samples, soil pit rather than cores, for available analysis already collected and archived? So do you accept soil samples already collected and archived?
So as part of MONet, no, not right now, but we encourage you to reach out and talk to us. We have other avenues. So we have our standard user program. The reason for not accepting them is not because we don't want them. It's just part of our standardization pipeline. So if we start accepting archived soils from all over the place, different depths, data becomes less comparable. And so we do have a full user program, and we're happy to facilitate that sort of access.
And if you are wanting to reach out to the MONet team, it's just as easy as sending us an email at monet@pnnl.gov. All right, so we can take another audience question if anybody has one for us. Yes, wonderful. Or, oh, I'm sorry.
You can take it.
Okay.
Hello. My question is about training to use these data. So you have this, all these new components, molecular components of data, and so we're democratizing data collection from everybody. How do we ensure that everybody has training to use these?
So I'll take it a bit, and then John and Emily can chime in as well. So one of the beauties of having this as part of EMSL and our partners with JGI and NEON is we have inherent in our user facility capabilities training. So this example for this summer, our summer school is focused on quote unquote demystifying multi-omics through data science. And so the whole intent will be how to use these multi-omics data sets and process them and visualize them and analyze them, which can be directly applicable to then taking the data from MONet and working with them. And I know JGI has a similar type of activity as well. And so, as Yuri said, you also have access to all of the software that we've developed, both at JGI with the metagenome analyses and annotation, as well as EMSL software suites as well.
All right. Was there a question over here? Yeah? Okay.
Thank you. So not that you all need any more work, but you know, in the way that the NEON samples have been an incredible asset, right, for a lot of folks, I'm curious whether once you get all these cores back, is there an effort to archive the samples and in a way hopefully get to share them later with folks that might have interesting additional questions that they would like to ask?
So right now we're not archiving cores, just partly because of it dramatically expands the cost scope of the project in terms for the archiving. However, a lot of the cores are actually collected from NEON sites. So in fact, we actually do have either direct replicates that are being stored by NEON, or we have very close approximations of them.
All right, another question from, oh, I'm sorry.
Linda, I have to add, to NEON does have, I mentioned the biorepository, I showed it. We do collect soils, dried soils, and archive those in the biorepository from all of our sites. 3 times a year, actually, we collect frozen soils that are then stored in liquid nitrogen for metagenomics and other sorts of resources. So there is a lot of soil there. We also partner with the NRCS as well, so they have a lot of soil. So, yeah, thank you. Yeah.
We've got, okay, 2 more questions from the virtual audience, and then that will be the end of this Q&A session. So, from one of our virtual attendees, amazing project. Yes, it is, thank you. All the credit here. Congrats to the developers. Are there any perspectives of an international partnership in the future? How is it planned long term? I'm sure we expected this question.
Yes, I guess I'll just say baby steps. The continental U.S. is quite large as it is right now, and so I'm not ruling it out, and we would definitely be interested in it, but I think our focus now is making sure we can tackle continental U.S., but we're happy to have conversations with potential international partners on how we might expand this more broadly, especially if we could standardize and open up those databases globally.
Yeah, and this one's another one about extending the network, but a little more locally. So this person would like to know, are there plans to extend the network to freshwater and seawater samples?
Well, I'll answer a bit for the one, because we also have, BER is funding a suite of projects that are involved in freshwater work. So we have our Compass project, which is looking at coastal water systems, as well as another open science effort that is a multi-lab effort called Wonders, that are looking at watersheds. And so we were trying to make MONet be complementary as opposed to duplicative. And so a lot of that work is actually already ongoing, and we're hoping to sort of just link into those databases as well. I won't, maybe I'll have John or Emily speak to the salt water, if we will. The coastal effort does do some of that, but it is not meant to be a true blue ocean.
So I guess I don't really have a lot to add to that. I think we are very fortunate in the sense that we have very strong collaborators right now through Wonders and the other projects that Douglas mentioned who are focusing on surface water collection. We're not absolutely ruling out that we'll be doing seawater sampling. Right now it is beyond, we're just focusing on trying to be successful in our first year with soils. Soils are really super complicated. And so that's really where we're putting our effort right now. So ask us again in another few years, and thank you for the question, whoever asked.
All right.
All right. Well, I want to thank all of our presenters here today. This concludes the MONet launch event, and I want to thank all of our attendees online. And for those of you here in the ESC lobby, we have a reception here afterwards that we encourage you to join. And I wanted to thank you all again and hope to see all of you working with us to make this MONet effort successful.